Good evening. My name is Mina Consuela Ruiz. I am currently cursing third semester of medicine, and today I'm going to talk to you about this original paper that is titled "A Novel Polymerase Chain Reaction Assay for the Detection of 7 Mycoplasma Species of Cattle Origin." Below, we can see the author's names and the publication date of the article. So, to get in a little context, we have to know what we are studying. In this case, the microorganism is Mycoplasma, which is a small bacteria with no cell wall that may cause some diseases as mastitis, arthritis, reproductive conditions, and respiratory conditions. There are many types of Mycoplasma. However, this study is centered on 7 of them. That is the Bovis arginini, Bovigenitalium, Californicum, Alcalescens, Dispar, and Canadens. So here we see a diagram of the mycoplasma structure, and on top we can see real mycoplasma through microscope. So we also have to get familiar with this experimental method, which we have already come familiar with through the COVID-19 test. This is an image which is a reference point because the basis is the same. It's an enzymatic amplification of DNA used to identify specific genetic material, in this case, mycoplasma's genetic material, right? So it could be used for many other things as we see here, but in this case is the detection of bacteria. This article's general objective is to develop a pair of polymerase chain reaction primers for detecting ruminant mycoplasma pathogens. And for this, there are a series of methods that could be divided in 4 big steps. First is the DNA extraction from standard isolates that begins with culturing isolates in mycoplasma culture bath at 37 degrees for 72 hours. Then the DNA is extracted by boiling the isolates, but the isolates differ from each other because there are different types of mycoplasma, right? So there has to be an adjustment of concentrations of the respective isolates. The second step, or second big step, is the sensitivity test, which begins with collection of bulk milk and milk from cows with mastitis. From the samples, they took 1 milliliter that was centrifugated at 3000 RPM for 10 minutes, and then they collected 100 microliters of the liquid supernatant, and the DNA from the milk samples was prepared as the AxiPrep bacterial genomic DNA mini prep kit protocol said. So the third big step was the primer design, which was based on the 16S rRNA gene sequences of various mycoplasma species. The chosen main sequence from the partial 16S rRNA was from the Bovirinus, that was 19 to 99% homologous to the Bovis. The most similar parts between both was compared with the region with the highest dissimilarity in other bacterial and animal genomes. Then the oligonucleotide sequences was chosen for the newly designed primer, right? This is 5' to 3', but the most important part from this step is the size of amplicon is 233 pair bases. That is going to be important in the results. So lastly, for the big steps is the optimization of PCR for mycoplasma, right? So this was performed in a reaction of a total volume of 20 microliters, which is a mix of stuff, which basically went through a process of denaturalization, then annealing, and then extension, right? So then the PCR products, if you want more information, it's in the PowerPoint, like in the slide. But then the PCR products were separated through electrophoresis and to present agarose gels and stained with ethidium bromide solution. Then it was visualized with a UV transilluminator, right? So these are the famous results that we can see figure 2 and figure 3. The most important thing about this is, for example, M is a marker for molecular weight, and we can see that on the bottom right, it says 233 BP, that is pair bases. That is the size of amplicon of this mycoplasma. So it's like the way of detecting that the mycoplasma is there, right? So here we see a series of lanes that 1 through 8 is the same as 9 through 16 in different concentrations. So we can see that some are more highlighted and some different concentrations than others, and that is why they had to make the adjustments in the methods, right? So here we can also see some PCR results from mycoplasma, and they're almost all highlighted. However, some are more than others, like for example, lane number 5, that is the mycoplasma arginine. They also had to demonstrate that they were not doing this randomly and this was not a random detection. So they did some more tests with microorganisms different than mycoplasma. For example, that Staphylococcus, we can see here the positive control marking on 233 pair bases. However, there are no other markings, which means that the specificity test is well. We see more of the same, the positive control and none other. That also corrects or validates the specificity of this PCR test. And then we see the sensitivity of the mycoplasmas, that is for the limits of detection, basically. So these are some others that were mentioned in the discussion, and they are all agreeing with what the results were according to the sensitivity and the lacks complexity of the experiment. And lastly, these are my own conclusions, that are that modern biological techniques such as PCR allow faster and less complex diagnosis, which may prevent further negative consequences, and that well-managed biological tools lead to a better understanding of small organisms such as mycoplasma, and thus permit a more efficient way to detect them and take assertive course of action. This last slide is my map, and I hope you liked it. Bye.